viewers of Biotechnica, welcome back again to another wonderful video. Today's video is going to be really interesting if you are someone who is thinking that I would like to know about how a vaccine is actually designed. Is vaccine designing a wonderful career to take off, or what are the future scope that vaccine designing has? So this is going to be the topic today. What's the future scope of vaccine designing? So come along with me and let's discuss the complete topic in detail. So let's proceed on with what is the future scope of vaccine designing. We already know there was a person called Edward Jenner, and he was the one who actually designed the first vaccine. We already know about smallpox, chickenpox, everything in detail. So first let's talk about what is the importance of vaccination and designing a vaccine. We understood the main role of vaccine is very predominantly seen when we were in the pandemic situations. Yes, we already know there are a lot of pandemic which has happened in the entire globe. So here, this time when we really had a testimony that we have seen COVID-19 which has come, and we saw how this vaccine is designed in a very faster way. So let's talk about the importance of vaccine designing or vaccine development. So what is this vaccine development? We already know there were situations that there were a lot of incurable microbial diseases, viral diseases predominantly occurring in the past many years. And when vaccine development has come back, there is a greater medical intervention that has taken place, and because of these vaccines, it just stops the transmission or reduce the incidences of many future pandemics. Yes. So when we talk in case of mumps, or when we talk in case of Zika virus or Ebola virus or many virus, we have manufactured a lot of vaccines. Even in COVID-19, we have produced many vaccines. Covaxins we have produced. So we produce vaccines, and in order to prevent the further existence of pandemic anymore. So that's the purpose of this vaccine. And if you talk about vaccine designing as a career or vaccine designing as one of the field, its annual saving of at least 2 to 3 million lives is saved worldwide because of vaccine, which means vaccine is definitely going to be a wonderful prevention techniques which can prevent a lot of infectious diseases which can be applied for infectious biology, and many people are saved because of these vaccines. So when we have to talk about the different type of vaccine, we already know there are live attenuated vaccine, killed whole organism vaccine. We already know what's a vaccine. Vaccine is actually an inactivated form of an antigen given into the human body in order to trigger the immune responses by producing antibodies. That's why we saw the first exposure and the second exposure. The second exposure, the immunological response is maximum. So we know about the live attenuated, the weakened or inactivated vaccine. Some of the examples: measles, mumps. Previously when we talk in case of MMR, which is measles, mumps and rubella, which all the three diseases together, we used one vaccine, which is MMR vaccines we used. And this is mainly a live attenuated vaccine where we weaken the viruses and introduce into the human body so that the human body produces antibodies naturally. This was for smallpox we have done. Then we improved to kill the whole organism. We just kill the whole viral organisms, and then we introduce into the body mainly for the typhoid diseases. And toxoid vaccines also we used. We used mainly for diphtheria and tetanus infections. And subunits vaccine, we have taken protein part like purified protein parts, recombinant protein, polysaccharides, very subunits from the virus we have isolated, and we treated for pertussis, influenza. So all this has been used, but it was first used in anthrax. Virus-like particles we used for human papilloma virus. Initially, it was used for hepatitis, and outer membrane vesicles, protein polysaccharide conjugates, viral vectors, nucleic acid vaccines. Of course, DNA vaccines are, when we talk in case of SARS-CoV-2, it is actually a DNA vaccines. So we isolated the DNA. Example, SARS-CoV-2, we have actually used nucleic acid vaccine, which is DNA vaccines. And it was first introduced in SARS-CoV-2 only in 2022. Yes. That is a greater evolution that you can see. DNA vaccine has come into the market. Bacterioid vector vaccines are also done. It's been experimental till now. An antigen presenting cell vaccine is also experimental. It's been going on, so we have to wait for a moment to check how this works. And this is the stages that vaccine development pipeline takes place. When we talk in case of the previous vaccines like measles or mumps or smallpox, it took a lot of time for us to get the vaccines for the entire global world if we talk in scenarios. So first it started with isolating the epitopes from the antigenic determinants we isolated, and then we manufactured the vaccines, and then we went in for clinical trials with animals and then in the human sample, and then we brought them into market. So it started with the discovery phase, preclinical phase, small scale phase, clinical trial 1, 2, 3, and then evaluation discussion, and then we manufacture, and then we check in for safety monitoring phase. And almost it takes, you can see, almost 10 to 15, 20 years it takes to bring one vaccine into the market. But if you imagine during SARS-CoV-2, we have manufactured vaccines a bit faster. How this actually happens? This is mainly because of the bioinformatics tools, or the omics technology has taken control in this scenario, and because of that we were able to sequence the proteins or RNAs or DNAs very, very faster. So vaccine designing, when we talk in case of classical times, classical vaccine designing will take a lot of time for you to do, but because of the invention of this omics technology, we were able to design the vaccines a bit faster. So if anybody is interested in going in for vaccine designing, my personal suggestion is going to be, you definitely need to know some bioinformatics tool. You need to learn omics technology. So I'm going to talk that also in detail. Okay, now the next scenario is vaccine development. What's the current status and future needs that we have to go in for? So I'm going to talk about the statistics. So nowadays pharmaceutical companies, they started innovating next generation vaccines. They started producing different types of vaccines these days. So let's talk about some of the vaccines. Like for autoimmune diseases, they started using cancers, allergies, infectious diseases, many diseases. Even for Alzheimer's disease, we started doing vaccines for this one. So which means whatever disease you're talking about, vaccine is playing a very important role, very, very prominent role, I can say. So over 260 vaccines are in development for the prevention or treatment of diseases. This was a report by 2017 update. You can see medicines in development using vaccines. So biopharma companies started using more than 260 vaccines focusing on the prevention and treatment. This is based on America's biopharmaceutical company, which means we believe that more vaccines are going to come. This is the current status, and it's going to increase even in the future decades also. So vaccine development and vaccine designing is the most crucial thing for doing research and bringing something novelistic drug into the market. So vaccine is very wonderful. Now let's talk about the third point, which is vaccine development and responses. Yes, so how a vaccine is actually developed and how it goes inside the body and brings about the responses. So first let's talk about the vaccine development. So I'm going to show you the complete how it has actually been designed, and I'm going to incorporate some of the omics technology also. So first we're going to identify potential proteins for inclusion in vaccine. So we have to understand what are the proteins that are present in a very specific virus or bacteria very specifically. So virology, bioinformatics is very, very important. If we have to talk about VBRC, nerve, everything. And after that we will determine which vaccine platform will be used, whether it's going to be DNA vaccine or subunit vaccines, or whether we are going to use a live attenuated vaccines or viral particle vaccines, whatever it is. So what type of vaccination am I going to produce? That is the second thing. And next we have to understand or determine transcriptional unit. Yes, we have to know what are the transcriptional units by using some of the promoter scans, polyeprid we will use, and we will find out which promoter to use, which enhancer to use, polyadenylation site, everything we'll be talking. And the next one is optimize several aspects of transgene. We have to optimize certain conditions that you can learn it when you go for research. And later we will understand or determine the protein expressions, very, very specifically the antigen expressions, by introducing them into an animals in the laboratory condition, and we'll understand what is the protein expressions that is taking place that you have produced in the laboratory. And then you will check for the immunogenicity in the rat or animals. and you will check for the protective efficacy. What is the efficacy of the antigen that you have produced? And finally, you will go for the trials, very specifically for human trials, and then we will finally bring them into the market. This is how a vaccine will be developed, starting from identifying a protein, and then what type of vaccine you're going to prepare or manufacture, and then some transcriptional factors, and you have to go in for some conditional changes like codon usages, Kozak sequences, and splice size, what you're going to check in for, nucleotide repeats, everything will be checking. And after that, we will go in for the protein expression levels in vitro assays. In the laboratory, we'll introduce into the animals and we'll check the immunogenicity and the efficacy of the antigen. And if everything is fine, then we'll go for the clinical trials 1, 2, 3, 4. And then finally, if everything's safe, secure, quality, everything is checked, then we'll introduce into the market. Okay, now when we're going to introduce into the human individual, what is going to happen? So there's going to be primary responses and secondary responses. All of you know about it. So primary responses is like the body is going to encounter the antigen for the first time. So which means the body starts the mechanism of producing lot of neutrophils, macrophiles, and B cells, T cells, everything will be produced. And we know that the memory cells usually records the antigen that comes inside the body during the first exposure. Suppose the second exposure, the same antigen is coming into your body, the memory cells remembers it and simultaneously produce all the beta cells and produces the antibody against, and it's going to be faster and large amount of production of antibodies are going to take place. This is how we have increased the responses by using vaccination. Primary responses and secondary responses are going to be kind of long-term responses to vaccinations. So vaccine development and responses is definitely very, very important. Without understanding this, we can actually never design a vaccine at all. Next one is designing vaccine in the era of genomics. So how this genomic has taken control, why we were able to manufacture these vaccines a bit faster than the previous years. So you can see it over here. I'm going to talk about the genome sequencing. Yes, we know about genome sequencing. We started using NGS. So reverse vaccinology we have. So I'm going to show you an image which talks about the complete things here. So you can literally see we use UniPort mainly for the proteins, and we are going to identify, if you're going to talk in case of a bacteria or viruses, extracellular membranes, outer membranes, and even virulence factors. We can understand what are the virulence factors over there. And after these membranes are localized and secreted virulence proteins. So if you're talking about a virus onto the outer surface, what are the virulence proteins that are present? We're going to identify. And after that, we're going to identify the molecular weight, the antigen, transmembrane helicases from the primary sequence that we got. We will be using BLAST, PDB softwares, everything, and we will find out the 3D structure and protein-protein interactions of them. We will find out, and then we will find out the epitope. That's the most important thing, because antigen is going to have an epitope. This epitope goes and binds to the paratope of the antibody. So if we know the epitope, which is an antigenic determinant, then we can easily find out, or we can introduce into the human body and manufacture a vaccine. So we have to identify B cell epitope productions. So we will use some softwares here, Epidocs softwares we'll be using. MHC we can go in for, and we can also calculate the affinity, MHC binding affinity calculation, and virulence of predicted epitopes also you can find out. So the main purpose is, because of these bioinformatics, we were able to predict the epitope of a very specific viruses. So screening of selected proteins, epitope production. So started with pre-screening, then we'll go for selecting the viral protein, which causes antigenic nature, and then epitope predictions. After doing epitope predictions, you can see here. Then we will go for safe. We'll introduce into the animal bodies, small animals like rats, and then we'll introduce into the human trials, and then FDA approval, and then recommendation, and then it comes into the market. So epitope based vaccine design is very, very important. So analyzing the epitope of the antigen is very, very important. So when you go in for designing a vaccine, this is a most crucial thing, where we'll start off with antigen selections from proteomics database. That's what I showed you. BLAST we saw, so UniPort we saw. So we use all those techniques in vitro studies, and then we will go for T cell, B cell epitope selection. Just now I showed you how it is actually done. And after that, we will do an in vitro testing in animals to find out the immunological responses in that animal, and then we will finally test it in the human individual. So in vivo animal experiments, and then we'll introduce into the clinical trial, and then we'll go for this one. So when you want to design anything, vaccine very specifically, epitome based vaccine era, which means we incorporate genomics technology here, omics technology here. So if you know all these things like docking, if you know pre-screening techniques, if you know screening of predicted proteins or epitome production using all the softwares, it becomes very, very easy for you to go in for research if you are interested in designing a vaccine for a particular diseases also. So let's talk about the next point. So this point talks about how bio-nanotechnology can play a major role in vaccine designing. So we have different things like self-assembled proteins and peptide scaffolds. Yes. So scaffolds are something like matrix, so it's going to interact with a lot of proteins. So self-assembly of viral structural proteins and other proteins. So when we're going to do a antigen, when we're going to manufacture a vaccine for from a virus, very specifically, we used to take the viral proteins, whichever is present over there, and we will take up the other proteins. So assembly of other proteins and viral proteins, and we will create a nanostructured scaffolds. And this can be used as a potential vaccines also. Next is bacteria-derived scaffolds. So one example is T6SS sheath, which is a scaffold. And this is also used for, it's a heterodimer of Vibrio cholerae proteins, and this proteins can be used as an antigenic determinant for introducing into the human individuals to find out it. And lipid-based scaffolds also we started using. We use lipid nanocapsules like ICMVs we started using. So when we have to talk about bio-nanotechnology, we started using a lot of scaffolds from outer membrane vesicles like lipids, lipoprotein mimicking nanodisc, dendrimo nanoparticle scaffold, just now I told about the bacteria-derived scaffold. So we started using bio-nanotechnology even in vaccine designing also. So if anybody is interested in vaccine designing, get to know bioinformatics, the omics data, as well as some bio-nanotechnology things. So that is going to be very, very useful when you go in for this. And of course, the cloning thing is definitely an important matter. Of course, NGS, everything you have to study. Next one, advantage of using certain fields in vaccine designing. Okay. I already told you we're going to use vaccine omics very specifically for vaccine designing. That's why we were able to manufacture a bit faster. So what about SARS-CoV-2? SARS-CoV-2 is no exception because we produce a DNA vaccine using this omics only, genomics. So SARS-CoV-2, you can see we have isolated variants of concern and variants of interest. Of our interest, we have isolated, and we went in for multiple sequence alignment using the sequencing tools, and then consensus sequences only we isolated, which means allied repeated sequences we have taken, and then we also checked in for some mutation has taken place or not. Then we went and identified the antigenic proteins that are present in the virus. And then using this softwares, we started using epitope prediction, as I already shown you, B cell and T cell epitope predictions. And then based on this epitope prediction, we started designing the vaccines. And then we went for in silico studies and physiochemical characteristics. And then we introduced into the animals and we checked, and finally we produced them as a vaccine into the market. So it made a bit faster just because of the genomics techniques that we're going to have, the bioinformatics tools that we have. So we made it a bit faster. So now let's talk about what the advantage of using certain fields in vaccine designing. So first is proteomics. Of course, as we already talked about genomics, proteomics plays a very important role. So for serum proteins, yes, abundance, antigens we are going to isolate. So definitely proteomics plays a major role, and in order to determine the structure of it, in order to determine the function of it. And metabolomics, yes, if you wanted to go for a subunit vaccines, you will be going in for lipid or glucose polysaccharide isolation. So serum metabolites, very, very important. So metabolomics plays a major role in vaccine designing. And as I already told you, immunoinformatics like finding out the HLA, B and T cell receptors, very specifically we talked about the epitopes, and antigen prediction and designing happens with the help of informatics only, and system analysis, and then genomics. Yes, we already talked about the transcriptional factors you need to understand using this genomics, epigenomes, microbiomes, or VAS. So this gives us a very important understanding whether it's going to be for designing a vaccination. You need to solidly have a knowledge in all this omics technology, which is going to be a great help. So when we talk in case of SARS-CoV-2, also we talked about that. We isolated the variant of interest and variant of concern, and we have taken the sequence using this NGS sequencing, and then we have consensus sequences, which means the aligned repeated sequences we looked in for, and we also check in for mutation, and then we went in for epitope predictions like B and T, and then we designed a vaccine, went for in silico studies, and then we checked in into the animals, and finally we manufactured the vaccine discovery. So if I have to talk about the different fields, proteomics, metabolomics, immunoinformatics, genomics also plays a major role in vaccine designing also. Okay, now what are the new technologies that has come which made a lot of improvement in vaccine designing? Yes, we started off with live or attenuated vaccines and whole killed vaccines we have used. Now we have come to DNA vaccines very specifically. So live attenuated vaccines were used before for smallpox, chickenpox and MMR, and nucleic acid based vaccines, which we can say it's DNA vaccine. Now we have the SARS-CoV-2 has been synthesized. mRNA vaccines, it takes very less amount of time to manufacture mRNA based vaccines, and it does not have a live virus, so there's no disease risk of causing any kind of disease in a person. So even COVID-19, some of the COVID-19 vaccines are designed using mRNA vaccines, and subunit vaccines are like you're taking only a polysaccharide or a conjugate vaccine or subunit vaccine, which was used for all these things like hepatitis B, human papilloma viruses, whooping cough, and virus-like particles based vaccines. Here we started using it for they do not have a genetic material. Virus-like particles do not have genetic material, so it's very, very effective human vaccine, and it's predicted that 90 to almost 100% it is very, very effective. So virus-like particle vaccines are very, very helpful, and we have non-replicating viral vaccines also. And we started using new vaccine delivery technology also. We have DNA vaccines, as I already told you, SARS-CoV-2, and it's easy and inexpensive. DNA vaccines are very easy and inexpensive, and it has a very long-term immunity. And when we started using new vaccine technologies also nowadays, how we are going to deliver is whenever initially when you talk in case of vaccine, we used to introduce through the injections. So nowadays we have alternatives to injections, like we started giving transcutaneous injections, and we started taking transgenic edible plants that contains the gene for the human vaccines. We started eating transgenic edible plants also, and we started using delivery depot systems. When we have to deliver a vaccine, we started encapsulating those vaccines with biodegradable polymers like lactic glycolic acids, PLGA, everything. Why we started using a different delivery methods? Because when you're going to use only injections, there are a lot of side effects. So we started improving on the technologies to introduce or to deliver these vaccine into human individuals, because it decreased the reliance on repeated injection. You don't have to keep on repeating the injections. You just have to eat a transgenic edible plant, or you have to take this transcutaneous things only. So nowadays, when we have to talk about vaccine designing, there's a great advancement when we talk in case of making a vaccine designing or introducing those vaccine into the human individual has made a bit very, very advanced than before. So it's amazing to take up vaccine designing as a research career also. Designing the next generation of vaccine, we started using, not only that, nowadays we started using, previously we used some antigen and adjuvants only we started using, and we introduced vaccinations, and we will look for the short term and long term responses. Nowadays we started using antigen, adjuvants and amplifiers, which actually amplifies the amount of antigen that we used, and we are going to introduce it over there. And based on this comparison, classic and amplified vaccines are considered to improve the responses to vaccinations. So this is going to be the next generation vaccines that we started producing with the adjuvants and amplifiers. And what's the future of vaccine going to be? Yes. So the main purpose of the vaccine is to prevent or to stop the spread of diseases when it comes to pandemic situation. Not only new vaccine we are going to produce, we started improving the old vaccines that has very less efficacy. So that's the most important thing. We started producing new vaccines for diseases which is not able to treat at all, and we started using, or we improved the vaccination technologies in order to improve the efficacy rate of the vaccines by using certain technologies. And we have used certain innovations nowadays. improving the simplicity and efficacy of vaccine delivery. As I already told you, we started using some deliverable materials, and we started using transgenic plants. So reducing the need of multiple doses. One problem previously we had is when you're going to take a vaccine, you have to give many times the same doses. Multiple doses has to be given for the body. But now what we have done, if you take one time or two time doses, very less amount of doses, it can literally boost your immune responses. So we started improvising in the vaccine designing also, and improving the manufacturing processes. As I already told you, mRNA vaccines are very, very easy to manufacture. So same way we started improving the manufacturing process in different ones, and we started delivering the vaccine doses very quickly. And we made even transportation and storage of these vaccines also a bit easier in order to reduce the wastages. So we have made a lot of improvements in vaccine designing and vaccine development also. But if I have to talk about, does vaccine designing is going to be a future scope? What is the future scope they are going to have? Yes, it's used in different field for application of novel generations of vaccines. So I'm going to show you this. So those who are really impressed by vaccine designing will love to hear this. The rising problem of antimicrobial resistance. We have a problem that antimicrobes are showing resistance against certain vaccines. So we can apply vaccine designing a bit improved in this kind of field, very specifically when we talk in case of Acinetobacter baumannii or Pseudomonas aeruginosa. So these has certain resistance against certain antibodies. So we have to improvise on that. And emerging infections and old pathogens. Since a lot of infection keeps coming and old pathogens are still existing, immortal in nature, this vaccine therapy is definitely going to be a very important one. Vaccine designing will always be there ever. And people are at risk because elder people are there. And even there are people who are having this immunocompromised patients. They are having a less immune system. Until these things are prevailing, of course, vaccine designing will definitely be there. So it is, of course, going to be a greater future scope if you take it as a career also. And new prophylactic and therapeutic vaccines are coming in the market. So definitely vaccine designing is important. And we started developing vaccines again, non-infectious diseases. We not only bacterial infections or viral infection, we started developing vaccines for cancer, diabetes, Alzheimer's disease. So which means vaccine is playing a role in non-infectious biology also, in infectious biology also. So vaccine designing is really, really important when you go into any kind of diseases. So vaccine against biological weapons. Yes, when we talk in case of biowarfare or biological weapons, when we use DNA vaccine, we can literally overcome those things. And new vaccine delivery technology, as I already told you, we started using, instead of using only injections, we started, without compromising the efficacy, we started using different methods of delivering the vaccines. And what are the challenges we still now face? So we have a lot of future scopes. Of course, vaccine designing is having a future scope. But do we have any challenges when we go in for this vaccination? Yes. Access to vaccines. So we don't have a lot of access to vaccine everywhere in every part of the world. And anti-vaccination movement is there. And commercial viability, we are not getting it because lack of vaccine for some diseases are there. When we talk in case of no effective vaccines are also there. There is no vaccines at all also there for some commercial diseases because there's no commercial incentives for the development. And we have a knowledge gap in designing a vaccine. That's the biggest problem. That's what I'm telling you, that everybody has to know about the omics technology, all these things. If you know this, then the knowledge gap will be reduced actually. And no effective vaccine is available. When we talk in case of tuberculosis, malaria or HIV, even though we improvised a lot, but not very effective vaccines are still existing till now. So until these things are there, definitely vaccine designing is definitely going to have a greater future in the upcoming rate. So I believe that this video is definitely going to be a great help if you are looking for research in vaccine designing or vaccine development. So I believe that this is helpful. So if you really like this video, please like, share and subscribe to our channel. This is Caroline Green. I'm going to meet you back again with another video.